My name is Russell Carter, and today I'm representing Group 2 lab technicians, and we're going to be talking about freezing HeLa cells, or CHO cells. First, we're going to go in and we're going to prepare our reagents and put them in the hot water bath. Then we're going to clean the hoods. Then we will check the cells to see whether they're confluent. If they're 80 to 100% confluent, then we will prepare to freeze them down. We're going to wash them with 1x PBS, then we'll trypsinize them, and then we'll incubate them, put them in the CO2 incubator for 5 minutes. Afterwards, we're going to check to see if the cells detached under the microscope, and then we will immediately add media to the trypsin in order to dilute it. After which, we will take an aliquot for the cell count, then we will put 9 milliliters in a falcon tube in order to centrifuge it. Once we're done centrifuging the cells, we will resuspend them. We'll discard the supernatant and resuspend the cells in the MSO media. Afterwards, we will place our cells in a labeled cryovial and place it in Mr. Frosty. And then once they're frozen, we need to leave them in the negative 80 degrees freezer for at least 4 hours in Mr. Frosty. Then once they're frozen, then we will put them in the liquid nitrogen and update our log and clean and be done. Okay, so this is my friend Joseph Ramirez, and he's going to go through the motions with you today, and he's going to go in there right now and get the trypsin and the DMSO and the media and put it in the hot water bath.
All right, let's get started.
So now he's going to clean the hood so that we can start working with our cells. And note that he's wearing his sleeves so that he doesn't contaminate anything with his lab coat sleeve. What he's doing right now is he's removing the cells from the CO2 incubator. Then he's going to check the color of the media. And what you would want to do at this point is record the color of the media in your notebook and take a picture of it. Then now he's going to check the cells under the microscope to see that they're confluent and they're not contaminated with anything. Mycoplasma is a huge contaminant in the lab, and you want to check to make sure there's no hair or anything like that. And then he's going to move it to the hood where he will wash it with 1x PBS. Then he will add trypsin and place it in the CO2 incubator for 5 minutes. So after 5 minutes of trypsin, he's going to remove the cells from the CO2 incubator and check that they're detached under the microscope. Taps it on the desk to make sure they're detached and check it. Now he'll clean the flask and place it in the hood in order to add media to the flask immediately. So after he aliquoted the cells for counting and for centrifuging, he's going to now bring the tube over to the centrifuge in order to spin the cells down while someone else does the count on the hemocytometer. So what he's going to do now is remove the supernatant and resuspend the pellet in 1 milliliter of 10% DMSO media. Then he'll transfer that resuspended pellet into a labeled cryovial. Okay, so you want to have your Mr. Frosty set up so you can take your cryovial directly from the hood and place it in the Mr. Frosty. And afterwards, you're going to put the Mr. Frosty in the -80 freezer. We're going to document in the cell lines book where we're going to put our cells. You need to make sure that you choose the proper Dewar number and that you choose the proper cane position and that you document properly exactly what is on the cryovial and the date when you added it. That's your Dewar. That we did the written documentation of where we're going to put the cells, we have to do a digital documentation. All the instructions on how to access the digital documentation is right here on the computer. So now that you did that, you're done, right? You fool! You didn't clean the hoods!